Hello and good afternoon, everybody. Welcome to this month's Genomics England Research Seminar. My name is Gillian Hastings-Ward. I'm the chair of the participant panel at Genomics England and a participant in the 100,000 Genomes Project. I'm delighted today to have two speakers. We have Dr Liz Ormondroyd and we have Dr Eleanor Seaby. Both of these ladies have been working on the data which is in the 100,000 Genomes database. Ellie is going to come first and she's going to talk to us about a gene-to-patient approach, uplifting novel disease gene recovery and identifying 18 putative novel disease genes. And then Liz is going to come on and tell us about her work on the additional findings in the 100,000 Genomes Project. Correlation with healthcare impacts. Sorry, I'm just going to move this part of my screen because I've got too many pictures going. Correlation with health conditions, impacts on recipients and healthcare utilisation. I've got a little bit of housekeeping to do first. And again, I'm going to have to juggle my screen in order to make that possible because I didn't print off the sheet. I'm sorry. So just bear with me a second and I will get that up. The main things to tell you are that this session today is being recorded. So anything that you ask as a question or anything you put in the chat may end up being public on YouTube, so please bear that in mind in your comments. We will be able to upload the webinar itself onto the Genomics England YouTube channel afterwards, and that will be then able to be viewed by whoever wants to come along later on. There'll be time for you to ask questions of each of our speakers today, but please, if you can do that by putting them in the Q&A box rather than the chat box, it'll be easier for us to find them. And we're going to give each of our speakers an uninterrupted 20-minute break period to speak, and then we'll have a few minutes for questions for each of them after that. If you run out of time for answering questions within the time available today, our speakers will be able to answer your question directly in the Q&A box. If you put it in the Q&A rather than the chat function, please. That would be great. And I believe either of them would also be happy to hear from you with any follow-up questions using the emails that they're providing in their presentation slides afterwards. Other things to let you know just now are that this is the final seminar for 2022. There will not be an event in December. It'll be the final Tuesday of December. We'll all be too busy having Christmas holidays, so there isn't going to be another one until January. And the details for that will be issued quite soon by the GSIP team at Genomics England. So without further ado, I'd like to introduce Dr Ellie Seaby. As mentioned, she's looking at novel disease gene discoveries, and she is a clinical research fellow in genomics informatics, completing her PhD joint supervised by the University of Southampton and the Broad Institute of MIT and Harvard. Her research focuses on novel disease gene discovery and methods to uplift diagnosis rates for patients with rare diseases. I would like to remind everybody to put your questions into the Q&A function, please, and let's hear from Ellie.
Thank you very much. I'm just going to find my slides. I think can I just check that looks okay?
Yes, that's great. Thank you.
Perfect. So thank you very much. So yes, today I'm going to be talking about using a gene-to-patient approach to uplift novel disease gene discovery, which has identified 18 putative disease genes. And it's an absolute pleasure to be speaking today. So as most of us know, there are about 20,000 human genes. And at the moment, about 6,000 of those are disease genes. And of course, it's possible that there are many more human genes that could be disease causing. The problem is that the function of most genes are unknown. So the difficulty is that it's very resource intensive and expensive to go and test every single gene and work out the biology of the gene to then find out what happens when you mutate the gene. So this can become a challenge when we're thinking about discovering novel disease genes. So why are finding novel disease genes important? Well, at the moment, across all types of genetic diseases, on average, about 30% of patients that have a predicted monogenic disease receive a diagnosis. So there's clearly a big room for improvement. And often with these patients that we see in clinic, we're pretty sure that they have a genetic disease, but we can't find their diagnosis. And the thing is that until new disease genes are discovered, diagnoses can't be made. So we need to find the genes, get them added to the gene panel so they can then be tested for in clinic, and those diagnoses can be returned. But of course, this is challenged by the fact that we don't know the function of most genes. So how can we identify novel disease genes? Well, some genes are more tolerant to loss of function mutation than others are. And what I mean by that is that for some genes, we can tolerate being a homozygous knockout. So many of us will go around and will have complete knockout of both alleles of a gene, and it will have absolutely no consequence. Now, this doesn't necessarily mean that those genes aren't important, because you can get gain of function mutations. But in terms of loss of function, for many genes, they're completely redundant. However, on the other hand, there are some genes that are completely essential. So what I mean by that is that you can have one tiny little variant or mutation in that gene, and it can cause a severe disease. Some of them might cause death in utero, and some of them might cause a very severe phenotype. So what we can do is we can use reference population data sets like GNOMAD and TopMed to help us understand the essentiality of genes. And we can do this using gene constraint. So what we can do is we can model mutation constraint by looking at the number of observed loss of function variants compared with the expected. So say for a given gene, you might expect to find 10 loss of function variants. Well, let's say you only found 1. That would tell you that that gene is constrained for loss of function. It's intolerant to loss of function, because people that have a mutation in that gene are not then reproducing, and therefore those variants are getting purged out through natural selection. On the other hand, there are some genes that are very polymorphic, and you can tolerate mutation quite easily. So maybe you'll find more loss of function variants in those genes than you'd expect just by chance alone. And that's where something called LUFF comes in. So it stands for the loss of function observed over expected upper bound fraction. And it's said like the egg in French, so LUFF. And what this does is it identifies genes along a spectrum, so a continuous spectrum of constraint for loss of function. And what we know is that genes with low LUFF scores are intolerant to knockout. So you observe fewer loss of function mutations than expected. And these are more likely to be disease genes. And this is helpful because what it can do is it gives us the potential to predict disease genes even in the absence of known function. So if we know a gene is intolerant to loss of function, what we can hypothesize is that if you have a mutation in that gene, that it's likely to cause a phenotype. And we know that known disease genes are enriched for low LUFF scores, and I'll show that in a minute. And this is just showing you the LUFF score here along the bottom. It ranges from 0 to 2. So genes that have a low LUFF score are intolerant to loss of function and more likely to be disease genes. And genes that have a high LUFF score are tolerant to loss of function and are often what are considered redundant genes. So what you can look at LUFF and you can compare it with what we already know about disease genes. So this down here, these come from the LUFF flagship paper, the GNOMAD flagship paper in 2020 by Kaczewski et al. And here we've got the LUFF decile on the bottom. So LUFF has been binned into its deciles. And we've got the proportion of genes against their LUFF score that are in OMIM. And what you see is that genes with the lowest LUFF deciles, the lowest LUFF scores, have the highest burden in OMIM. So they're more likely to be disease genes. And then it falls down, and then you get this little peak here. And this peak is because of recessive genes, where you'd expect them to fall along the middle of the spectrum. And in fact, this along here shows the same thing. So here we've got gene lists of known haploinsufficient genes, known autosomal recessive disease genes, and known olfactory genes. And when you plot these against the LUFF decile, what you see is that haploinsufficient genes are highly enriched for low LUFF scores, and they tail off rather rapidly. Then you have autosomal recessive genes, which are going to show a degree of constraint, but not as much. They fall around the middle of the spectrum. And then these olfactory, highly polymorphic genes are at the end of the LUFF spectrum. So you can clearly see how the LUFF spectrum does show quite nicely the difference in disease gene types. And in fact, if you look at the first LUFF decile, 35% of those genes are already disease genes. They are already known characterized disease genes. So this then made me think, well, there could be up to 65% of these genes that are intolerant to loss of function that may well be disease genes. And this was really the target of where I wanted to look in my research and see, well, could we find patients that maybe had variants in these 65 novel genes not yet associated with disease and try and identify novel disease genes? So the methods, what I wanted to do was to apply what I'm calling a sort of genotype to phenotype approach. So traditionally, when we think about analysis of data, we start with a patient, we sequence them, and we end up with lots of variants. And the majority of them are going to be benign, but then maybe you have one disease-causing variant. And when we're using gene panels, if that gene panel does not include the gene of interest, so the gene where you would have your disease-causing variant, which would be true for a novel disease gene, then you often miss that diagnosis, or you miss that novel disease gene. What I wanted to do was to flip that on its head and do what I'm calling a genotype to phenotype approach. So here I'm looking for genes with low LUFF scores that I think are pathogenic, loss of function variants because they're the most damaging to an allele, and then assigning those variants directly to patients. And what I'm trying to find is, are there patients that have loss of function variants in the same gene with overlapping phenotypes? Because that's going to increase the confidence that these could be real disease genes. So I use data from the 100,000 Genomes Project, and I just really want to say that we're so fortunate that we have such a rich data set, such as the 100,000 Genomes Project, to allow us to do that. Because for ultra rare diseases and for rare diseases, you know, we need large data sets to be powered to detect patients that have these rare variants. So what I did was I looked in the genome data of about 14,000 parent-child trios, and I looked for rare, de novo loss of function or predicted loss of function variants in genes with a LUFF score less than 0.2, so in that first LUFF decile. And I was only interested at this point in looking for genes that were not already associated with disease. And then I was most interested in identifying patients that had loss of function variants in the same gene with overlapping phenotypes. So this situation that I was describing over here. And the results, which you can see in my paper in Genetics and Medicine earlier this year, what it shows is that from about 14,000 trios, when you apply this very simple filtering step, we identified 643 loss of function variants in LUFF-constrained genes. Now, 475 of these were in known OMIM genes, and the majority of these, in fact, 98% of them, were actually pathogenic variants. And that's part of a separate paper. But looking at the novel cases, we found 168 variants in 126 unique novel genes. So I then asked myself the question, well, of those genes, was there more than one gel kindred affected? For 99, the answer was no. So in 99 cases, we had 99 loss of function variants in 99 unique genes. So these were still interesting, but not as interesting as the cases where we had multiple individuals with loss of function variants in the same genes, but with overlapping phenotypes. And what we observed was we had 18 genes where we had multiple families with similar overlapping phenotypes, all with loss of function variants in those genes. And that gave us 18 putative disease genes to begin with. And the way I defined a disease gene was looking at OMIM, and this was done back in 2019. And when I started looking in more detail at some of these genes, I found that 5 of them had been published within sort of a couple of months of my analysis. So these hadn't been indexed yet in OMIM. So I'd essentially identified genes that I thought were pathogenic, thought were disease-causing, that then actually proved to be. And in fact, the patients in gel matched, or the phenotypes of the patients in gel overlapped with what was in the literature. So automatically, my method was I was proving my method worked. And then this left me with 13 putative novel disease genes. And these are the results. We've got the 13 here along the bottom. And now since 2019, 6 of them have been published. So they have all been case series with functional experiments that are published in decent journals. And what's interesting and exciting is that the gel patients, many of them have now got a new diagnosis in these genes. But of course, there's still some that haven't been published. And so what I've been doing is I've been working on those genes. And I'm going to discuss 2 of them today. So hdlbp and Ddx17. And there were, so with hdlbp, we had sort of 2 with predicted loss of function variants, 1 with the start loss, with phenotypes so these were the HPO terms that were overlapping between the patients, and then Ddx17, these were the overlapping phenotypes. And we're now building case series and doing functional experiments. So Ddx17 is an RNA helicase, and it controls processes very important in early neuronal development. And opening up our search to Matchmaker Exchange, we've identified 12 patients globally so far that have a de novo variant in Ddx17 with a neurodevelopmental phenotype. And about half of them are loss of function and half are missense. And all the patients have sort of a global developmental delay, delayed speech and language development, intellectual disability, problems with fine motor delay, and autistic features. There are other features that are shared between them, but those are the sort of key ones. And what we're doing is we're working with the European Xenopus lab at the University of Portsmouth, and also working with a group in France to try and prove that our hypothesis that if you mutate this variant, if you have loss of function, haploinsufficiency of Ddx17, it causes a neurodevelopmental phenotype. And I just want to show you some of the work that's been done by my colleagues at the European Xenopus lab. So here we've got some control tadpoles along the top, and here we've got 2 guide RNAs where essentially we're injecting at the 1 cell stage some crispants, some guide RNAs to cause homozygous indels. And what you see is in a lot of cases, you get these severe gastrulation defects. Now, some of them are milder, and these do go on to survive, but the majority of them have severe gastrulation defects and don't survive. So of the ones that do survive, what the group did was they started to look at some measurements. So very early on, so this is the control, so this is early stage and late stage, and this is the crispants, early stage and late stage. And they measured some distances. So they measured the lens to lens distance, they measured the distance between the eyes, and they also measured the brain length. And early in development, what you can see is that there is a statistical difference between those that are controls in black and those that are crispants in blue. But later in development, as they continue to develop, you don't see that that size difference, that sort of normalizes out. And actually, that's quite interesting, because in the patients, the majority of them don't have dysmorphic features. So they don't look dysmorphic facially. I'm not sure if this is going to work. Hopefully you can see. So they've done an assay where they've looked at the movement of the tadpoles. So you can they can track them using this clever technology, and then they can use that to determine how well the tadpoles are moving. And you can show in light and dark conditions. So here we've got dark conditions, here we've got light conditions, and blacks are the controls and purple are the crispants. And what you see is that the controls move more, and when you go from light to dark, they become startled and they sort of wake up and they start moving more. And overall, the crispants, they move less, and they don't have this same startle response. We've also been very fortunate to work with a collaborator, Julien Crochet, in France, and he is working on some experiments in mice, and he's been looking at neuronal development in early stage mouse brain. And what he's shown is that Ddx17 is involved in axon development. So these are controls here in white, and these are mutants. And there's a statistical difference in the axon length and the number of branches in the brain of mice in terms of neuronal development. And again, that's shown here. There's reduced branching, and there is a difference in axonal length. And we're now repeating those experiments in Xenopus. And what we're also doing is we're building an F1 model, a heterozygous Ddx17 knockout model, and repeating our assays. And then the other gene I'll quickly touch on is hdlbp, which is an RNA binding protein, which has got high brain expression. And so far, we've got 27 patients globally that have a de novo variant in hdlbp. And again, this is showing a developmental phenotype with speech and language problems, intellectual disability, fine motor delay, autistic features. So very similar, but also we see some craniofacial abnormalities as well and some feeding difficulties. And again, we're modeling hdlbp and Xenopus and working with a group in Germany who are modeling specific variants for us. And again, similar experiments. When you, at the 1 cell stage, when you inject the crispants, what you see is a high burden of homozygous indels in Xenopus tropicalis. Embryos are essentially lethal early in development. So they're just unsurvivable. So what they then did was they injected 1 of 2 cells. So at the embryo stage, there were 2 cells. They injected 1 cell with the crispant and left the other cell wild type. So what you're trying to do is to create a sort of mosaic of hdlbp. And these are the uninjected controlled tadpoles along the top. And here we've got our crispants along the bottom. And what you see is that there are craniofacial abnormalities in the crispants. There's also some difference in left to right facial symmetry. Some of the crispants have defects affecting the esophagus. And this is sort of this area here where you've got this sort of green area. A number of tadpoles have scoliosis, and you can see that in the tails. They do look slightly bent. And also the crispant tadpoles are generally smaller. Now, we're unsure if that's because they are smaller sort of from a craniofacial perspective, or if they're just smaller because they're not developing as well and they're not feeding as well. So what they've done is they've done some micro CT imaging as well of, again, one of this 1 of 2 cell embryo stage. And what you can see between the controls and the crispants is that the crispants have some abnormalities in the organization of the branchial arches. And there's also this sort of slightly odd downward projection of the head, sort of here, which is different to the controls. And there's ongoing work looking at that at the moment. And then in terms of movement, the other thing that you can observe is that in black, you've got the controls, and in red, you've got the crispants. And what you see is that crispants are moving significantly less than the controls. And you're seeing this in the patients because they have delayed walking, they have delayed movement, delayed fine motor development. And again, we are also doing, we're building an F1 model to test this in a heterozygous state. So that's just to give you a sort of flavor of what I've been doing. And really, it's credit to the fact that we have these large data sets, such as the 100,000 Genomes Project, which is helping us identify novel disease genes. And so far, 12 of the 18 genes that were identified in 2019 have been published, and which is helping return diagnoses to GEL patients. And then we're working on these case series for 6 remaining genes, and we've got functional work going on in 5 of them at the moment. So, of course, I'd like to thank you for listening, but most importantly, the patients and the families enrolled in the study, the European Xenopus Lab, my entire supervisory team, and all of my collaborators. And that's a real, this isn't just me, this is really a testament to how science moves forward when people work together. So thank you very much for your time.
Thanks a lot, Ellie. As a non-scientist, I think I managed to follow most of that as well. So congratulations. We've got 5 questions have come into the Q&A. So we'll just start at the top, and it may be that you've answered some of these questions in the course of your talk. So if that's the case, then just set us straight. But the first question I have is there any mechanism to explain disease genes with high LUFF scores? Absolutely. So a really good example of this is a gene called EHH-ADH, which is important in mitochondria. And this has a really high LUFF score, and you can tolerate complete homozygous knockout and you have absolutely no problems. However, it is a disease gene because if you have one particular missense variant very early on in that gene, it causes a gain of function, and essentially that's the mechanism that causes disease, and it causes a Fanconi syndrome. So there are some genes that if you mutate them in a you change the way that gene works and it gives it some kind of advantage, then you can have a disease mechanism that way. So it's without it you're okay, but if you give it an extra property that then can interfere with something else, then that can cause disease. So that's where we see it most, is gain of function, where you can have a very high LUFF score, but still be a disease gene. Thank you. Question 2, how do you determine whether you have a true loss of function without experimental study?
Yeah, again, it's a great question, and it's difficult, and so we don't always get it right. But what we've tried to do is use as much manual curation when we're looking at our loss of function variants to actually try and prove that they really are loss of function. So we're doing things like removing them if they're in the terminal exon. We're checking to see that there's no splice rescues, that they're not, you haven't got, it's not an indel and then you've got a rescue indel that's in frame later on. So you can use those kind of skills to try and curate the variants to say that they are most likely to be loss of function. But it is a great question. And really, without RNA sequencing, you can't truly determine whether they are loss of function. But I think the fact that there's so many patients that have now had return diagnoses with phenotypes that are overlapping with those that are in the literature supported by functional work, there clearly is, enough evidence to say these are disease genes.
Thank you very much. I'm just going to throw in another cheeky one before we get to the next one in the Q&A. And it's a question from me. My son has a gain of function variant in his gene. And I just wondered whether you're thinking about looking at gain of function variants at some point, or whether you're focused solely on loss of function.
So at the moment, my focus has been on loss of function, but I think, in the future, absolutely, I think it is a really interesting area. And I think that maybe with as we start to try and unravel these variants of uncertain significance, and we can have high throughput ways of modeling them, perhaps with protein modeling, then maybe we'll be able to understand gain of function variants a bit better. But primarily I've been working on loss of function, but obviously very happy to, to work on different things in the future.
Thank you very much. Okay, back to the Q&A. Lovely work there, says Jamie Ellingford. There's two key assumptions in the cohort level analysis. One, we can predict loss of function variants accurately. And two, we have an accurate estimate of the expected number of loss of function variants per gene. He wondered whether you could comment on the limitations of the approaches applied.
Yeah, absolutely. So again, a great question. So completely true that you have to be able to accurately predict loss of function variants. And often we don't. As I say, not every variant that is called a loss of function variant really is a loss of function variant. And this is still a space that's evolving, and there's lots of work going on to help us truly determine. So one way you can look at it is you can say, in a population of healthy people, how many of them have a loss of function variant in a gene that you'd expect to cause a severe phenotype. And then that might help you understand reasons if that really isn't a loss of function variant. What are the properties about that means that it's not loss of function? And that can help us learn. So it's absolutely true. We need to be better at determining what's loss of function, but that's still an ongoing process and is difficult to automate it. Because there's so many areas that need to be thought about. In terms of the second part of that question about the accurate estimate of the expected number of loss of function variants per gene, again, it's a great question. And, we can't, it is a prediction because you can't actually count what the actual number would be. One of the limitations is sometimes gene length can play a role in that. So if you have very large genes and very small genes, especially for small genes, it becomes harder because the events are much rarer. And in fact, their LUFF score only gives a score for about 19,000 genes. There's still 1,000 genes that don't have a LUFF score for that very reason. So the question's really good. And of course, there are limitations such as, we need more people. So if we had, a huge magnitude more people in population databases, that would give us a much better understanding. So that will help in the future as more people are sequenced, we have more population data sets, and also that our ways of annotating and understanding and curating loss of function variants improve.
Thank you very much. Next question, using LUFF to prioritize truncating variants and prioritizing disease genes is great and has helped in discovering many new diagnoses. Do you have any brief thoughts on how gene prioritization when it comes to missense variants?
Yeah, so there's been work where people have looked at missense constraints. So across a gene, some genes show particular missense constraints. So it might be there's a particular area in a gene that's particularly constrained for missense. So if you have a missense variant at the beginning of the gene, it's not so important. If you have a missense variant at the end of the gene, it might not be important. But bang in the middle, maybe there's a really critical phosphorylation site or something very active about that particular protein, that if you mutate that specific point, then it can cause more disease. So there are missense constraint models. And one thing I've been looking at doing is doing a similar thing, but saying, let's look for very highly missense constrained regions of genes and then look for missense variants in those regions and do a similar thing. So that's one way that you might be able to prioritize missense variants. And then also you can use in silico prediction tools as well, which can be helpful in conservation. But I think these regional missense constrained tools will be really helpful in that regard. Thank you.
Now, I see we're going to run out of time for most of the rest of these questions, so I know that you'll be able to answer them in the A function of the Q&A in a second. But just one more verbal before we move on, if you'll forgive us, Liz. Someone says, thank you for your talk. Is it a coincidence that most novel genes found were associated with intellectual disability, or was this cohort from a specific subset of the population of the gel data?
It wasn't from a specific subset, but there's two reasons that I think this could be the case. One, there are a lot of intellectual disability cases in gel as a general rule. And secondly, when you're looking at loss of function variants in these highly constrained genes, they often have quite important functions. So it's not that surprising that they're not necessarily going to be just specific to one organ, that they're going to have a severe phenotype and potentially affect the brain, affect early on in development. So I think those are the two things that come to mind as to why perhaps you see that. But of course, we need a more diverse data set with lots of heart disease and kidney disease and various other things to see what the distribution really is.
Okay. Just to add one thing, sorry, I saw there was a question about additional diagnoses that you'd made in known genes, and just to say that yes, Ellie's made lots of diagnoses for 100,000 Genomes Project participants. Thank you.
Thank you very much for that, Lisa. And thank you very much, Ellie, for your talk there. I'll leave you to get on with answering some more of the questions in the Q&A, and thank you again for agreeing to do that. And we will turn our attention now to Dr. Liz Ormondroyd. Thank you again for joining us today, Liz. Liz is a principal investigator in the Radcliffe Department of Medicine at the University of Oxford. Her research since 2005 has focused on ethical and practical delivery of genomic technology, in particular how different types of genomic information are used by patients and families and in clinical care. Liz originally trained in molecular biology and then as a genetic counsellor. She's been in Oxford since 2008, and before that was a senior research fellow at the Institute of Cancer Research. She's been involved in the 100,000 Genomes Project since its inception. And she's here today to talk to us in a talk entitled Additional Findings in the 100,000 Genomes Project, Correlation with Health Condition, Impacts on Recipients and Healthcare Utilisation. And again, thank you very much for joining us, Liz. You have 20 minutes and then we'll open the floor to questions. So if anyone has any questions for Liz, please put them in the Q&A. Thank you.
Thanks very much for that introduction. And it's a pleasure to be here. I'll try and share my screen. Can everyone see and hear? Okay.
Yes, that's worked. Thank you.
So yeah, I'm going to be talking about the study we call a SAFE study, which is looking at outcomes of disclosing additional findings from the 100,000 Genomes Project. I'm going to talk about the background and rationale for the study, the methods, and some very preliminary results, but we're still collecting data, so we haven't really started to really analyse the data yet. So, as I'm sure you don't need reminding, the 100,000 Genomes Project recruited around 85,000 participants through the NHS between 2015 and 2018. Participants could be adults or children with a rare disease that had a suspected genetic cause or cancer and some healthy relatives. Participants provided blood samples for genome sequencing, and ascertaining patients who met eligibility criteria and recruitment by a variety of healthcare professionals was a huge undertaking for the NHS. The project was a healthcare and research hybrid initiative, so it had clinical aims, but consent for research was integral, and participants agreed that their clinical and genetic data would be stored and used for ongoing research, as we've just heard. And there was also optional consent for additional findings. So additional findings are, so participants were offered analysis of a defined set of genes which are known to be associated with health conditions that often don't present until they're at an advanced stage, and for which there are evidence-based risk management and interventions available in the NHS. Additional findings are unrelated to the sequencing indication, and the vast majority of participants elected for that analysis. The focus over the next few years was in genomic analysis for the primary condition, and analysis and release of additional findings happened later than planned, but most have now been released. So Genomics England do a preliminary analysis and then release data to the Genomic Medicine Service Alliances, where the NHS regional genetics labs check that variants are pathogenic, confirm presence and report to clinical teams. Participants are then informed through agreed letters whether they have a finding or not. And then clinical teams use a variety of means to support, inform and direct clinical care. Just to explain at this point that the terminology can be a bit confusing, so I may use the term secondary findings, and that applies to those genomic findings that are not believed to be related to the presenting condition. Those would be primary findings. Additional findings are the name that Genomics England uses for the specific secondary findings that they look for and report, and I may use both. This is a screenshot of the additional findings question from the adult rare disease consent form. So you see that participants were offered a binary choice. They were told that conditions might be relevant also for their family, and that they could develop a condition even if an additional finding was not reported. But you see that it doesn't mention genes or conditions specifically. There was a list on the 100,000 Genomes Project website, but you see one of the points says that the conditions might change anyway. So, and anecdotally, it was quite challenging to counsel about this particular question and to ensure that consent was informed. This is taken from the Genomics England guidance document from last year, and these are the genes that are screened for additional findings. So this is a short list of genes for which there is good evidence for implication in causation of or predisposition to the disease shown in the third column. All conditions are cancer predisposition or familial hypercholesterolemia. The guidance provides not only for the genes, but also for the variant classes within the genes. And the aim was to report only variants with a high probability of being pathogenic. And some of these conditions can be important in childhood, or there are risk management actions relevant in childhood. But for others, predictive testing is not generally offered in childhood, and these genes are only screened in adult participants. And just to go back a little bit and think about secondary findings. So they've always been controversial. The landscape changed in 2013 when the American College of Medical Genetics and Genomics released a guidance document, recommendations for reporting incidental findings in clinical exome and genome sequencing. And this recommended that pathogenic variants in a number of genes should be sought and reported in all individuals undergoing sequencing. The guidance has been updated by a multidisciplinary working group, and the latest iteration shown here lists 78 genes, which includes the Genomics England additional findings genes, as well as others, a large number of others, including inherited heart conditions, which are not, apart from familial hypercholesterolemia, not in the GEL list. Concurrently, and in contrasting position, the European expert group, the European Society of Human Genetics, put forward recommendations that within a healthcare context, genome sequencing should focus on the original test indication, and that analysis should be as targeted as possible. And that position was unchanged in 2021, as you see from the paper on the right. And I think a summary is that the ethical principles of proportionality, autonomy and justice should be considered. So I think it's fair to say the debate is ongoing. Over the last 10 years, there's been a lot of research published focused on the ethical and social implications of secondary findings and the views of a range of stakeholders. For example, our 2017 systematic review included 44 papers, mostly from the US, and found views on a spectrum. And increasing knowledge, either among patients and the public or healthcare professionals or other experts, tended to result in a greater appreciation of the complexities and development of more cautious views and favouring an approach limited to returning high confidence variants. But stakeholders generally supported return of actionable findings, even though actionability definitions did vary. So why is it important to study outcomes of disclosing additional findings? This has been called a genotype first approach, which contrasts with the well-established phenotype first approach, for which there's considerable evidence about a range of impacts to inform clinical practice. So in the more traditional phenotype first approach, current guidelines recommend testing in certain clinically affected individuals, those with a strong family history or both. But obviously individuals who don't meet those criteria are not offered testing. A key issue is what's the penetrance of pathogenic variants in the population, so the probability of disease development given a pathogenic variant. Current estimates of penetrance for most gene variants are based on genetic testing in families affected by the disease in question. But penetrance estimates from family studies can't necessarily be applied to the general population. And there may be shared genetic or environmental factors that increase or decrease risk. And I just include here a quote in the title of a paper from 2018, not pathogenic until proven otherwise. There are now studies, including some looking at disclosure of secondary findings to biobank participants, which find that current penetrance estimates may overestimate risk in the general population, although it's important to note that we don't know a priori whether the people that who are receiving additional findings, whether they have a personal or family history of that condition. So penetrance estimates drive clinical decisions, such as risk-reducing surgery, but there are many other actions that patients and clinicians might make. So, for example, the frequency and age at commencement of screening, drug prescription, compliance, family planning, reproductive genetic testing, life planning, and telling relatives. Identifying people who have a pathogenic variant but whose risk of disease is low could lead to unnecessary diagnostic procedures, surgeries with possible complications, as well as anxiety and cost. And because penetrance can be age-related or is age-related, surveillance may need to be over many years and sometimes lifelong. The experience of receiving secondary findings may differ from receiving results in a predictive genetic test setting, which we're a lot more familiar with. In that situation, patients have consciously sought specific genetic information. They're likely to have greater familiarity with the condition and to have prepared psychologically for receiving the results, and as well for the clinical actions that would be recommended if they did have the variant. And also results are available in a defined and usually short time frame, 2 or 3 or 4 weeks. Whereas in a genotype first setting, such as the additional findings setting, a concern a few years ago certainly was that there might be psychological harms which might impact decision making, as well as creating a need for additional psychological support. And we have no idea whether people remember any discussion of additional findings during the consent process, or what they understood, or whether they remember their choice, and they may be notified about an additional finding at a particularly vulnerable time. So there could be benefits and harms, but since this is new activity, not only for the NHS, but more widely, we don't have a good understanding of the benefits and risks. So in the SAFE study then, we wanted to collect evidence to understand some of these issues, which might be important to inform policy around additional findings. And just to note here that we're only looking in the SAFE study at health-related findings. We're not including carrier status. So we want to understand what's the risk of disease, what's the evidence, is there any evidence of disease associated with the additional finding in the individual or their family? How does the NHS respond? What referrals do they make? What tests and clinical screening and risk management interventions are offered? How do individuals respond? What meaning does this genetic information have for them? What actions they take and why? If patients do or don't have experience of the additional finding associated condition, how does that affect the impacts and decision making? And bearing in mind that recipients of additional findings already have another health condition that they know about, and that's the reason that they are in the project, either in themselves or in their family. And we wanted to look at the resource use and cost implications. The funding we have allows studying one genomic medicine service alliance, Central and South. The mechanism for accessing data was much discussed in setting up the project, this study, and in particular with Gillian Hastings-Ward on behalf of the participant panel. The 100,000 Genomes Project consent did not cover what we needed to do, and we didn't feel it was appropriate to seek research consent during the disclosure appointment. But on the other hand, it was important that we were able to understand and collect data from as many additional finding participants as possible in our region without recruitment bias. I didn't say, but these are quite rare. So probably 1 or 2% of people are going to have an additional finding. So we developed an opt-out process, which has confidentiality advisory group approval. So the clinical team sends what we call a patient notification document. It's just a two-sided piece of paper to all patients seen. And they can then opt out via a secure web link or by phone. And then we won't collect any data. Otherwise, we collect data from medical records and the research environment. Also, patients are asked by the clinical team if they're willing to be approached about a research interview, which uses open-ended questions and an interview guide informed by literature on impacts of genetic testing. And finally, we're using the clinical data we collect to understand resource use. And this just summarises the study and shows the research elements in green. And just to be clear that the patients we interview are a subset of the total number of additional findings recipients.
Sorry, Liz, can I just ask a question on that,
yes.
on that flow chart, please? The clinic appointments that you're talking about, would you be able to skip that? Thank you. The clinic appointments there, are they solely for patients and participants who've had a positive finding? They have had a result that shows that there is an additional finding for discussion, rather than for other people? They're all positive results.
So this is only, sorry, yeah, this is for the disclosure appointment.
To tell them the result of that.
Yes, so they're notified by letter, and sometimes they might have a telephone call, but then they're invited to a clinic appointment. So that's what's meant there.
Thank you.
So as I said, we're still collecting data, but I think we're kind of seeing the end point, at least for the sort of disclosure data collection. And so we have a total of 65, and we expect about 25 more. And you can see that the main conditions associated with additional findings here are breast ovarian cancer predisposition and familial hypercholesterolemia. We have small numbers from the other disease groups, and you can see that there's a range of ages. So most patients, but not all, have had a first appointment to disclose and discuss the additional finding, and we're working with the clinical teams to access data. So we're collecting data from the disclosure appointments, and for a minimum of 6 months later to try and get that kind of, what happens in the next period after the disclosure appointment, for all patients that we can do that for within the study timeframe. And we're triangulating data from medical research, we're triangulating data from medical records with the research environment. But there is a time lag with the research environment, so the data there at the moment is up to the end of March, and we're eagerly awaiting the next data release in January. And then we've interviewed 22 patients so far, or recipients of additional findings, and we have a reasonable representation by sex and age. So in summary then, we will finish data collection and analysis in the next few months, and maybe you'll invite me back next year to share the results, which we hope will inform the clinical utility of additional findings and future policy around not only whether but also how to offer additional findings, if that is in fact the decision. So we've got lots of people to thank, primarily Josh Nolan, who's done most of the data collection, but also clinical and laboratory teams at the 4 sites, Gillian, and all patients, in particular people who've given up their time to do an interview. I'd also like to thank our funders. Thank you very much.
Thank you very much, Liz. That was a really interesting talk. We have a question in the Q&A, which is, did you manually review all the filtered variants in secondary finding genes, or did you use an automated approach? I'm not sure if that's more a question for the Genomics England team who were actually returning the additional findings, or whether it's one for you, Liz. But
no, I think it's
definitely nodding in acknowledgement there. So perhaps between you.
Who's nodding in acknowledgement? Annalise, yes, it's
Annalise,
definitely not one for me. I think my role and the team's role is very much kind of studying the clinical processes rather than actually making those decisions. But if Annalise would like to say more about that.
I'm not an expert on the additional findings, but I think there was an automated pipeline, but then locally it will have been reviewed manually before return.
Yeah, I mean, that is definitely the case, that the regional genetics labs have gone through all of the variants. So not all of the ones that were released by Genomics England have ended up as being additional findings.
So the manual step is done locally.
Yes.
Thank you. Next question, again, it's for Annalisa. Do you plan to add polygenic risk scores to the risk estimation on top of the mutations? So in addition to the single gene additional findings, is Genomics England thinking about looking at polygenic risk scores too?
This could be a big question and probably off the topic of additional findings, in the sense that I think, as you were really clear about, the additional findings is a subset even of a much bigger list of potential secondary findings that might be used in other countries like the US. So I think it's been a more conservative end of the approach. Yeah, obviously from the research side, there's a lot of interest in polygenic risk.
Yes, thank you. And that Our Future Health, an entirely separate organisation, are really focusing on that in their work to recruit up to 5 million people from across the United Kingdom and do just that. Next question we have, could you clarify what you meant by accessing medical data in the research environment and really awaiting the next release? Does this mean the research environment is linked to NHS data?
That might be a question for Annalisa as well. Yeah, so I think the clinical data is pulled through NHS Digital. Am I right there? And so what's in the research and the Genomics England research environment, which is for all participants, is what's in NHS Digital. So it's got secondary care episodes and tertiary care episodes. There isn't a lot of primary care data in there, which is slightly unfortunate for us because we can't get things like lipid measurements if they're not reported back to the specialist. Is that answering the question?
And just to add, yeah, that's true. There isn't primary care data now. There are ICD-10 codes from the hospital episode statistics. So if somebody is admitted to hospital, for example, then they have quite good coding. For outpatients, it will tell you just what type of appointment they had, neurology, but not why.
Exactly, and I think it's been really useful for us to be able to sort of have both sources of data, the medical records, where we get a lot more sort of context and specific information about episodes that have happened, and then we can just kind of, and we know the date that happened, so we can sort of match that up within the research environment.
Yeah, and I think to answer the simple question, does this mean the research environment is linked to NHS data? Yes, it is. People who consented to be in the 100,000 Genomes Project and the people coming forward through the Genomic Medicine Service now, the consent that they've signed to say that they're happy for this data to be shared into the NGRL, the National Genomic Research Library, says that it will be updated in perpetuity. And there's several regular release points of data during the year where that, in the latest set of medical records about us, is uploaded into that research environment for researchers to look at behind the secure walls that they have in place there. Yeah. Right, the final question we've got here is the healthcare utilisation, oh sorry, now we've got two. Is the case, yes, sorry, to come back to the original question about the research environment being linked to the NHS data. You've said, is this the case for all patients sequenced through Genomics England or just for 100k patients? Yes, it's everybody who's agreed to share their data with Genomics England. So that's GMS and 100k.
For the NGRL consent.
NGRL consent. Right, we've got two questions left and I think we'll be out of time. So penultimate question, is this a healthcare utilisation component of the study looking at the counterfactual, i.e. are you comparing the costs associated with and stemming from returning additional findings with the status quo of not using genomic sequencing to screen for these conditions? Liz?
I'm just trying to work out what the question is. We're comparing costs associated with and stemming from returning additional findings with the status quo of not yet. We're working with James Buchanan, and I think he's planning to do a comparison of the patients who receive additional findings compared with those who don't, and so the healthcare costs of both groups of patients. But obviously we will have a lot more data on the patients that have received additional findings. And so it's really trying to work out both the costs of disclosure, so that's kind of the lab costs in terms of the variant checking, and then the kind of clinical costs of the appointments and providing the genetic counselling support around the appointments, around the findings. But also looking at, so for example, if it was an FH additional finding, you might expect a typical scenario is that the patient will ask the GP to check the lipid levels and prescribe a statin. And I suppose the FH ones are slightly simpler, but for the cancer predisposition ones, there are often various different kind of referrals that are being made, and we're wanting to understand what those are, and then whether patients attend. Does that answer the question?
I think so, yes. Thank you for that. Right, the penultimate question is a fairly straightforward one. Can participants make requests for variants that they're interested in, or a summary of variants that they have from Genomics England? I can answer that one too, and the answer is no, unfortunately. There was the bundle of conditions that were being, or the bundle of genes that were being looked at for the additional findings, was set by Genomics England. But there is a list, I believe, which is available somewhere on the Genomics England website that says which genes were looked at this point. So it does give some people some reassurance about what was looked at and what wasn't. Annalisa, do you have any further update on that? Did I get that right? That in terms of knowing which additional findings have been looked for in the current cohort of results?
Yeah, so that's in a list on the website, which ones, yes. And obviously individuals are entitled to make a subject access request for the data that's held about them, but I think that's a bit different to this question, where I think somebody's asking about whether they can make a specific request for variants, say in a particular gene, and that's not.
So as far as I am aware, this has not been updated since July last year, but this is just the health-related findings. I've not included here the carrier status.
I think the list has hardly changed from the initial list. Yeah.
Okay, thank you. And I think the answer to the question, the follow-up question here about why that is, a lot of it's due to the confidence with which the clinicians and researchers are able to attribute pathogenicity to these variants. So they need to be really sure that that's something which is going to be likely to cause you a problem in order to return that finding to you, in short.
And also this was a list agreed with the NHS where there's then a clinical pathway for return of them. So we're not returning them directly to an individual who suddenly hears they've got X. This is all coming through healthcare where there are set pathways that have been agreed with the NHS.
Indeed. Thank you very much.
And
right,
I think, sorry, can I just add that?
Very
I
quickly.
think it was important to just sort of emphasise that there should be evidence-based screening and risk-reducing actions that can be taken.
Okay, thank you very much. I'm afraid we're out of time. There is one more question in the Q&A. I don't know if Annalisa might have time to answer that while I'm wrapping up here, but I just wanted to thank you very much for your time today, Liz and Ellie. Two very interesting talks there, and you can tell by the level of engagement we've had that you've got people's brains working this afternoon. So thank you again for your time today. This is the final seminar for this year, but Genomics England will be back again in January with another research seminar, bringing even more science into the wider world. So thank you all again, and Merry Christmas when it comes. And I think Annalisa is just going to finish typing that question up. So we'll leave the chat open for a few minutes, but thank you again for today. Goodbye for now.